much. Thanks for coming today. It's truly an honor to be on this side of the podium and a little bit weird. I've been coming to the Speakers Series since my first year of grad school 7 years ago, and I've had the pleasure of seeing many fantastic talks and many exciting pieces of research in this forum. So I'm truly honored to be here, so thank you. Today I'm going to be talking about a few different projects that I've conducted with collaborators here at UCLA, primarily my advisor, Marty Hazelton, also Christina Larson and Melissa Fallis, and a few others who are in the audience. And I'm going to be focusing on my research on cycle shifts, so shifts across the ovulatory cycle in women's mate preferences and their sexual desires more broadly, and also in their attractiveness. There we go. Okay, so for nearly all mammals, the brief high fertility window of the ovulatory cycle is the only time when sex can result in conception. This leads to two straightforward predictions from an evolutionary perspective. First, evolution will have acted on females to develop adaptations that will influence their sexual desires, their sexual decision-making, and certain other aspects of their social behavior contingent on their current fertility within the ovulatory cycle. Second, selection will have acted on males to detect cues of high fertility in females, to perceive these cues as attractive, and possibly also to adjust their behavior in the presence of females expressing these cues to increase their likelihood of securing potential reproductive opportunities with those females. And so today I've organized my talk into two sections addressing these two general predictions about the ovulatory cycle. And then at the very end, I'm going to be discussing some general conclusions and implications of this work, and also some unresolved issues and questions that remain to be addressed by future research. So I'll start by discussing my work examining changes across the ovulatory cycle in women's sexual desires. So first, some background. Way back in 1998, when I was still probably in middle school, Stephen Gangstad and Randy Thornhill conducted a study using a method that has now come to be known as the stinky t-shirt paradigm. So in their study, they recruited a sample of young men to come into the lab, and they measured all of these men for their fluctuating asymmetry. So fluctuating asymmetry refers to the extent to which the right and left sides of the body deviate randomly from perfect bilateral or mirror image symmetry. They also sent all of the guys home with a never-worn, clean, white t-shirt, and they asked them to wear it for 2 nights while sleeping and while avoiding any behaviors that might alter their natural body sense. After the 2 nights, they had the guys come back into the lab, and they asked a sample of young women to smell all of the t-shirts and rate them for their sexiness and their pleasantness. They also asked the women to report whether they were currently using hormonal contraceptives or were naturally cycling, and they asked them to report the last start date of their most recent menstrual period. The hypothesis that they had set out to test was one that was motivated by theories in evolutionary biology and supported by research on a number of non-human species. So evidence from a wide variety of non-human animals suggests that fluctuating asymmetry is a marker of underlying developmental stability, which refers basically to an individual's robustness against various disturbances that it encounters during development that could interfere with its development. And developmental stability itself is thought to reflect the presence of certain beneficial underlying genetic qualities. So the logic goes something like this. Most organisms come into the world with a genetic blueprint to build a symmetrical body. However, they encounter certain stressors during development that can lead them to depart from that blueprint. So those individuals who possess certain beneficial genetic qualities, by which I mean genes that promote healthy immune function, an absence of genes with deleterious effects, or a low overall mutation number, might end up developing more symmetrically despite those disturbances than an individual who lacks those protective qualities. And therefore, their symmetry will be a cue to their underlying genetic quality. And symmetry and other correlated aspects of an individual's phenotype also tend to be attractive to potential mates. And so a straightforward prediction that Gangstad and Thornhill could have made is that as men's fluctuating asymmetry increased, so as they became less symmetrical, women's ratings of their sense would decrease. So they would find their sense less pleasant and less sexy. But they actually made a slightly more nuanced prediction than that. So here I've plotted out women's average probability of conceiving as a function of their current day in the ovulatory cycle. And as you can see, there's a tremendous amount of variation here. Importantly, these are averages across hundreds of women, so this graph may actually downplay the amount of variation that an individual woman would experience across the cycle. So past research suggests that an individual woman's fertility might look something more like this, the aqua line. So basically a 0% probability of conceiving until they hit that very brief high fertility window, then they have a very high probability of conceiving, and then it's right back to 0 again. So throughout evolution, the costs and benefits of women's reproductive decisions would have had their greatest impact right in this high fertility window, and therefore it would be somewhat surprising if women's sexual desires remained perfectly stable across the cycle. So with this in mind, Gangstad and Thornhill predicted that instead of seeing a general preference for the scent of more symmetrical men, women's preference for the scent of more symmetrical men would depend on their current position in the ovulatory cycle and their current fertility. And in line with their predictions, they found that women who are currently in the low fertility phase of the ovulatory cycle did not show a preference for the scent of symmetrical men, but women who are currently in the high fertility phase of the ovulatory cycle did. So as men's fluctuating asymmetry increased, they became less symmetrical. Women's ratings of their scent attractiveness, which is just an average of pleasantness and sexiness ratings, went down. And they interpreted this pattern as evidence that women may have the capability to detect information present in men's natural body sense that would historically have alerted them to the presence of genetic qualities that could have impacted their children's chances of survival and reproductive success. And not only that, but this information appears to have the strongest impact on their sexual desires, their attraction, when women would have been most likely to conceive, and therefore when their sexual decision making would have had the greatest impact on their reproductive success. So Gangstad and Thornhill's study and other similar studies that followed after they had done that initial study eventually ended up leading to the development of the ovulatory shift hypothesis, which proposes that there will be relationship context-dependent shifts in women's mate preferences across the cycle. So specifically, it predicts that as women's fertility increases within the cycle and as they near ovulation, they become more attracted to characteristics in men that historically were associated with beneficial underlying genetic qualities. Importantly, however, they think that this cycle shift will be strongly moderated by relationship context. So women are expected to exhibit this cycle shift in their mate preferences when they evaluate men as potential short-term sex partners, a context in which they would be relatively likely to have sex in the immediate future and therefore could conceive with these men and pass on whatever beneficial genetic qualities they had detected. However, women are not expected to exhibit this cycle shift when they evaluate men as prospective long-term partners, a relationship context in which they'd be relatively less likely to have sex in the immediate future. So this first pair of predictions pertains to women's preferences for cues to genetic quality, or at least to genetic quality in the ancestral past. Now the second prediction of the ovulatory shift hypothesis instead pertains to women's preferences for cues that would have indicated in the ancestral past that somebody might have made a good long-term social partner and co-parent. So this would be cues like kindness, parenting skills, and a willingness and ability to invest material resources in her and in her children. So the ovulatory shift hypothesis predicts that women will not experience a cycle shift in their preferences for these long-term partner suitability qualities in any relationship context. And the idea here is that ancestral females could have benefited reproductively by mating with males who provided these non-genetic benefits at any time in the cycle and across a range of different mating contexts. So before I move on, I should note that fertility-related shifts in female mate choice have been documented in several non-human primate species, but it's unclear from the current findings whether these shifts serve the same kinds of functions as posited by the ovulatory shift hypothesis, or whether they serve other functions yet. So do women's preferences shift across the cycle? Since Gangstad and Thornhill's pioneering initial study, This area of research has really taken off. So there are now dozens of published studies that have examined cycle shifts in women's preferences for various characteristics in men, and still other studies that remain unpublished. However, these studies have been very methodologically heterogeneous. They've especially varied in the measures that they've used to assess women's mate preferences, and also in the methods that they've used to estimate women's current fertility within the ovulatory cycle. And they've also produced somewhat mixed results. So this suggests that there's both an opportunity and a need to do a rigorous quantitative evaluation of the findings in this literature. So as part of my dissertation and in collaboration with my advisor Marty Hazelton and Melissa Fallis, we recently conducted a meta-analysis of the literature on cycle shifts in women's mate preferences. We used a variety of different methods to identify over 70 potentially eligible studies, so there are a lot out there. And some of the methods we used, for example, were sending out data requests via the social psychology listserv, and we also posted an advertisement asking for data in the HBEST newsletter. We designed inclusion criteria that would allow us to retain a large number of those effects that we identified through our initial search in our sample, while still also taking measures to limit our sample to those studies that would provide a coherence test of the ovulatory shift hypothesis. So I'm going to skip over the details of these right now, but if you have questions, I encourage you to ask me about them during the Q&A. So even after we selected studies based on our inclusion criteria, we found that we were left with a sample of very diverse mate preference measures. So most studies in this literature use a measure like this, where they ask women to directly evaluate the attractiveness of male faces or some other male stimuli known to vary on some characteristic of interest. For example, here they're looking at facial masculinity. But some other studies in this literature have instead simply asked women how important or desirable a characteristic is in a prospective mate. And we think that this latter kind of measure might tend to elicit women's reports of their general preferences, and therefore might be a little bit less sensitive to the transient variation in mate preferences predicted by the ovulatory shift hypothesis. In addition, most studies in this literature have studied women's preferences for characteristics in men that are at least widely thought to have served as cues of genetic quality in the ancestral past, and so there are pretty strong predictions for those characteristics. But some other studies have examined women's preferences for characteristics that are not yet widely accepted to have served as cues of genetic quality in the ancestral past, and so predictions are weaker in that case. In order to balance the trade-offs of including the diverse set of mate preference measures that's been used in this literature versus limiting our sample to only those that would provide the strongest test of the ovulatory shift hypothesis, we decided to actually analyze two nested samples of studies. So this first sample included a large number of studies and a large set of very diverse mate preference measures, and we were using relatively relaxed inclusion criteria to see if there are effects, would they generalize and would they remain robust even when we kind of relaxed our inclusion criteria. And then in the second study, we used stricter inclusion criteria, and we limited our sample to only those studies using methods that we thought would provide the strongest possible test of the ovulatory shift hypothesis. And so this gave us a sample of 96 effects nested within 50 studies in our broad or relaxed inclusion criteria sample, and that included 12 unpublished studies. And this gave us an additional 68 effects in 42 studies, 10 of which were unpublished in our more limited narrow sample. We computed Hedges G for each eligible effect. If you're familiar with Cohen's d, then this is basically almost the exact same thing. It just includes a slight adjustment that reduces bias in small samples of participants. And in this analysis, G is the extent to which women preferred a characteristic more strongly at high fertility versus low fertility reported in standard deviation units. So for example, a G of 0.2 would mean that their preference was 0.2 of a standard deviation stronger at high fertility than at low fertility on average. And at least in psychology, we would conventionally interpret this as a small effect. We used a multi-level modeling approach. This relatively new technique offers a range of benefits over traditional meta-analytic techniques, including the ability to properly include multiple effects based on the same sample of participants in a single analysis, rather than choosing between those effects. And we weighted each effect by the inverse of its variance, And this allows effects that were measured with greater precision, such as those coming from larger samples of participants, to have more pull on the weighted mean effect sizes. And then we compared the weighted mean G in a short-term relationship context versus a long-term relationship context, and also versus an unspecified relationship context, which I haven't mentioned yet. So importantly, some of these studies, rather than specifying that women should evaluate men as prospective short-term or long-term relationship partners, simply asked women to evaluate their attractiveness, physical attractiveness, sexiness, sexual attractiveness, or something like that. And they made the prediction that they would still see the pattern of cycle shifts that we get in a short-term relationship context. So this follows actually pretty straightforwardly from the original ovulatory shift hypothesis. Basically, if rating a man as more attractive or more sexually attractive or sexy increases a woman's chances of having sex with that guy in the immediate future, then we should expect a pattern of cycle shifts similar to what we see in a short-term context. In other words, these kinds of evaluations might plausibly mediate their immediate sexual decisions. And so we predicted that we'd also find a pattern of cycle shifts here similar to a short-term context, but they might be a little bit weaker. So the first set of analyses examined cycle shifts in women's preferences for a number of different hypothesized cues of ancestral genetic quality, and most of them had something to do with symmetry, masculinity, or cues of circulating testosterone. So regardless of whether we use the more relaxed or the stricter inclusion criteria, we found a pattern of cycle shifts that was perfectly as predicted by the ovulatory shift hypothesis. So women did prefer these characteristics at high fertility more strongly than at low fertility. And this cycle shift was strongest in a short-term relationship context, intermediate in an unspecified context, that's the purple bar, and then totally absent in a long-term relationship context. Next, we conducted a series of more focused analyses to examine cycle shifts in women's preferences for specific cues of ancestral genetic quality. And I should note that a lot of these were underpowered because we had small samples of effects examining preferences for these individual characteristics, and some of them looked at cycle shifts only in one or two relationship contexts, so we couldn't actually compare across the contexts. Nonetheless, we think that this provides at least some preliminary insight into where we're getting robust preference shifts and where we still need more research. So for body masculinity and behavioral dominance, we actually did get the pattern of cycle shifts predicted by the ovulatory shift hypothesis. So women showed a stronger cycle shift in their preferences in a short-term than in a long-term relationship context. We found some hints at that pattern when we looked at facial symmetry, scent of symmetry, and vocal masculinity, but all of these effects fell short of statistical significance. So I would say that we just need more research here to be able to make any strong conclusions about whether the preference shifts are robust for these traits. But it is notable that some of the largest effects we observed in our whole meta-analysis were for scent cues of symmetry. We thought that was kind of interesting. So if anybody has ideas about why we're getting such huge effects there, I'd be interested to hear them. The analysis examining cycle shifts in women's preferences for facial masculinity provided partial support for the ovulatory shift hypothesis. So we did observe the predicted cycle shift in an unspecified context and not in a long-term relationship context, but we didn't find the predicted cycle shift in a short-term relationship context. I have a hunch that that might be because that sample included two studies that used a potentially problematic measure of preferences for facial masculinity, and after I moved those two, there was a small cycle shift there, but it still falls short of statistical significance, so I don't feel comfortable making any confident claims about the presence or absence of effects in that context as of yet. So I would say we probably just need more research on facial masculinity to find out whether this unexpected pattern is robust and in need of explanation, or whether it's just a fluke due to some of the idiosyncratic characteristics of the studies in our sample. And then finally, the analysis looking at facial cues of testosterone was underpowered, so we don't really know. The vote's still out there too. Okay, so the second set of analyses instead examined prediction 2. So it looked at characteristics that are thought to have indicated long-term partner suitability in prospective mates. And this included things like parenting skills, kindness, faithfulness. So consistent with the second prediction of the ovulatory shift hypothesis, analyses did not reveal a positive cycle shift in any relationship context or in either sample. And even though it kind of looks like there might be a pattern opposite of what we saw for preferences of cues to genetic quality, I wouldn't make too much of it because the scale is kind of blown up here. So really what we're seeing is just teeny tiny, almost null effects across the board. So basically women just aren't experiencing any cycle shifts in these preferences. Okay, so given that we found some cycle shifts that were predicted by the ovulatory shift hypothesis, an important question is whether publication bias can account for these effects. So publication bias refers to the tendency for non-significant effects, which tend to be smaller, to have a more difficulty finding their way into the published literature. And this can cause upward bias in mean effect size across published studies. So our analysis had included both published and unpublished studies. Nonetheless, if there's publication bias in the published literature, it could still influence the mean effect sizes in our analysis. Yeah.
Were the unpublished studies in general things that were under review but not published yet, or were they specifically studies that people had tried and failed to get published?
Oh.
Yeah, or never tried, yeah.
We didn't ask them if they had tried, so I can't say for sure, but most of them seem to be just unpublished for good. There was one study that actually switched publication status, so it started out unpublished and then was published, possibly because once they took a look at the data again, they decided that it was worth publishing. But I just coded that as published for our analysis. All the rest, I think, are still unpublished. Okay. So as is conventional in meta-analysis, we used funnel plots to check whether small effect sizes were underrepresented in our sample. So in funnel plots, effect sizes are plotted against their standard error, and that usually creates this funnel shape with more precisely measured effects zeroing in on the mean effect size, which is our estimate of the true effect size. If bias is present, then you'll usually see that there are missing effects over here in the lower left-hand quadrant of the plot. And those are small effects measured at low precision, so exactly the ones that we would expect to fail to reach statistical significance that are still missing from our sample. But this is just an example. So we plotted out all of the effects where we were predicting a positive effect. So all of the effects that were measuring cycle shifts in women's preferences for cues of genetic quality in an unspecified or a short-term relationship context. And analyses revealed no evidence of bias in the first broader sample, and just a hint of bias in the second sample. And so we followed up with a trim and fill procedure, and this will actually tell you how many effects it looks like you're missing, and it tells you where to impute them. So we went ahead and did that, and this caused a negligible reduction in the mean effect size. So it looks like publication bias is not accounting for the significant cycle shifts that we observed. Another important question that has recently come to the fore in social psychology is whether our results can be accounted for by p-hacking. So p-hacking refers to the practice of trying out multiple statistical analyses or different ways to configure your data and then reporting whichever one works. In other words, whichever one yields a p-value of less than 0.05. So obviously this is really bad. It's possible that researchers sometimes do this without even realizing. For example, they might decide to add in a covariate, and then voila, they get the effect that they were predicting, and so they might think, okay, now I did it the right way, so I'll report that one. So whether researchers are doing this on purpose or not, it doesn't matter. It could still be making its way into the published literature and having an effect on the kinds of effects that we're finding in our meta-analysis. So obviously, p-hacking would inflate the type 1 error rate and could possibly contribute to false positives in this literature. And several critics of the cycle shift literature specifically have recently questioned whether the abundance of positive findings in this literature could be the result of p-hacking. So for example, one researcher said researchers might have begun with a window of 6 fertile days, and in the case of non-significant results, conducted exploratory analyses that successively broadened the number of days and maximized the distinction between fertile and non-fertile women's preferences. And the same kind of concern led another researcher to say many or perhaps all results may have been created by exploitation of unacknowledged degrees of freedom or p-hacking. So obviously this is a very serious concern. I hope that researchers in our field aren't doing this, but they could be. And so we ran several different analyses to check for this practice. So first, as you probably, well, I'll go back. So as you probably got from that first quote, one of the major sources of concern is that some of the studies in this literature use high fertility and low fertility windows that are defined after researchers have collected the data. Usually they define them beforehand, but sometimes they do define them afterwards. And so this at least presents the opportunity for them to try out different high and low fertility windows and report the one that produces a significant difference between high and low fertility. So the first analysis we ran to address this concern was limited to those studies that, rather than using discrete high and low fertility windows, just treated fertility as a continuous variable, which eliminates the possibility for researchers to engage in this kind of p-hacking practice. And so, as you can see, even among these studies, we do find the pattern of cycle shifts predicted by the ovulatory shift hypothesis. There's a cycle shift in preferences for cues of genetic quality in a short-term and unspecified context, but not in a long-term context. And in case you're wondering why the purple bar, the unspecified context, is so huge, that's probably because that context included 3 scent of symmetry findings, and those, for whatever reason, just happened to be the largest in the whole sample. Okay, so second, we used a brand new technique that was just recently invented by the same people who brought our attention to the possible issue of p-hacking in the first place. So they suggest using what they call a p-curve to find out whether it looks like a set of research findings is the product of p-hacking or is actual evidence that we can trust. And so a p-curve refers to a frequency distribution of published p-values that are less than 0.05 for some set of research findings. And as I mentioned, it's used to detect selective reporting, such as what can result from p-hacking or from just publication bias. And they ran a number of different simulations, and they found that the shape of a p-curve, specifically its skew, so whether it's right or left skewed, can tell you something about the evidential value of those findings. Without getting into the details, basically, if they see that there's a high proportion of p's that are just underneath 0.05, that suggests that p-hacking is going on for 2 reasons. One, because if somebody is intentionally engaging in p-hacking, they'll probably stop running analyses as soon as they get a p-value of 0.05 because that's their goal. And even if they're not, if there is no true effect, it's easier to get a p-value of just under 0.05 than it is to get a p-value closer to 0. So on the other hand, if you see that there are a lot of p-values closer to 0, that's suggestive of a true effect and no p-hacking. And if you get a flat distribution, that just is kind of inconclusive. So we constructed 2 p-curves. These were both looking at all of the effects for which we were predicting a positive effect. So this could be the comparison between high and low fertility preferences in a short-term or unspecified context, or it could be the moderation effect of a short-term versus long-term context on this difference. And regardless of whether we looked at the p-values that the researchers themselves reported, which included some p-values based on one-tailed or directed tests, or whether we looked at the exact 2-tailed p-values that we recalculated based on their test statistics, either way, we are detecting significant right skew. And this tells us that these data do have evidential value, and there probably really is a true effect. And just as a point of comparison, that dashed line shows us what the p-curve would look like if there was a true effect, but we had very, very low power. So basically, you just need to know that ours isn't more flat than that. If it were flatter, that would be inconclusive, but it's not, and the chi-squared test is detecting significant right skew, suggesting these findings do have evidential value and there is a true effect. So in sum, our meta-analysis revealed strong support for the pattern of cycle shifts predicted by the ovulatory shift hypothesis. It does not appear to be accounted for by publication bias or p-hacking. And an important question that follows from this is, what are the implications for women who are currently in a romantic relationship? If their partner lacks the characteristics that they find really sexually attractive at high fertility, could this impact their satisfaction with their partner or with their relationship? So Christina Larson took the lead on 2 studies to address this question. And one of the studies included 41 women, the second included 43. In both studies, women completed 2 sessions at high fertility and at low fertility. And they also completed a series of luteinizing hormone tests to check whether or not they ovulated. So luteinizing hormone surges approximately 24 to 48 hours prior to ovulation. So if we see a surge, we know that they're going to ovulate near their high fertility session. And at both of their sessions, they completed questionnaires assessing how close they felt to their romantic relationship partner, how satisfied they were with their relationship, how committed they were to their relationship. And also in study 2 alone, we obtained a measure of how characteristics they thought certain flaws were of their partner. So do you think your partner is lazy? And we found evidence to suggest that cycle shifts in women's feelings about their romantic relationship partners depended on how sexy they thought their partner was. So the less sexy women rated their partner, the more distant they felt at high fertility relative to low fertility. And the more critical they were of him at high fertility relative to low fertility. In contrast, the sexier women rated their partner, the more satisfied they were with their relationship at high fertility relative to low fertility. But I should note that we only found that effect in study 2. So one possible explanation for this discrepancy between the two studies, both of which did measure relationship satisfaction, is that in the first study, we didn't specify any time frame that we wanted women to think about when they were evaluating their satisfaction with their relationship. In the second study, we told women that they should think about how satisfied they've been over the past 24 hours relative to usual. And so that might have been a better measure for picking up on the transient shifts in women's preferences and feelings about their partner. So interestingly, we found that regardless of how sexy women rated their partner, their feelings of commitment to their relationship did not change across the cycle. So in sum, this suggests that although women's feelings about their partner and their relationship might change a bit across the cycle, this doesn't seem to have a measurable impact on their commitment to their relationship or their intentions to stay in their relationship. Okay, so now I'm going to shift gears and talk about prediction number 2 by showing some of my research examining cycle shifts in women's attractiveness. So in non-human mammals, the high fertility phase of the ovulatory cycle is often accompanied by dramatic changes in females' social interactions with males and also in their social interactions with other females sometimes. And one possible explanation for what triggers these changes is that males and females have the ability to detect cues of fertility in females and then to adjust their behavior in response to those cues. So what about women? Do women exhibit cues of fertility across the cycle? And if so, how do others respond to those cues? So in some non-human mammal species, high fertility female scents appear to have a particularly potent effect on male behavior. For example, at least one study has found that male hamsters will attempt to copulate with dummies that have been rubbed with fertile female scents. And when I first was interested in fertility cues, I thought this was a fascinating finding because the olfactory information is so powerful that it's able to trump visual information, tactile information, and other stuff that might make a male think twice before attempting to copulate with a dummy. And so when I first was interested in fertility cues in women, I wanted to see whether maybe there are fertility cues present in women's natural body scents. A few early studies suggested that there were. However, this data was limited because rigorous hormonal methods for evaluating women's position in the cycle were not yet widely available. And so, with collaborators at UCLA, I conducted my own version of the Stinky T-shirt study. We had 41 naturally cycling women participate as scent donors. All of them reported that they were regularly cycling and they hadn't used hormonal contraception in the last 3 months. This is important because hormonal contraception typically works by wiping out a lot of the variation in hormones across the cycle that might drive the kinds of cues that we're interested in studying. And all of the women provided scent samples at high fertility and at low fertility, and again, they completed a series of luteinizing hormone tests to make sure that they ovulated near their high fertility session. To collect their natural body scents, women wore gauze pads under their arms for a period of 24 hours while following very strict behavioral guidelines that we designed to minimize the contamination of their sample by non-natural scents. So for example, we asked them to avoid wearing deodorant, antiperspirant, eating anything really smelly, etc. And we also provided them with a gift bag that contained fragrance-free materials to use in the shower and everything else that they would need to adhere to our very strict odor collection protocol. 112 men participated as scent raters. They completed a few different tasks, but I'm just going to present the main rating task for now. So all of the men rated all of the women's high and low fertility samples, And for each woman, they rated her high fertility and low fertility sample, which had been randomly assigned to bottles simply marked A and B for their pleasantness, sexiness, and intensity. All they were told is that they would be rating samples that had been worn by women. They didn't know that the samples were worn by the same woman, nor that the study had anything to do with fertility. And as predicted, men rated women's high fertility scent samples as significantly more pleasant and sexier than their low fertility samples. They also rated them as a little bit less intense, but that effect fell short of statistical significance. And when I did a regression model, it didn't account for the other two effects. So we were curious to see whether this effect is specific to men or whether women also think that other women's high fertility scents are more attractive than their low fertility scents. So we recently ran a replication with 20 women as scent donors and an additional 91 women as scent raters. And we found again that, like men, women also rate other women's high fertility scents as significantly more pleasant and more sexy, and they also rated them as significantly less intense than their low fertility scents. Around the time that we completed the first of these two scent studies, Marty Hazelton and I were invited to conduct a review of the then just emerging literature on fertility cues in women in order to address the central question, can men detect ovulation? So this is a graph that we presented in our paper, and it shows effect sizes from representative published studies at the time. So in general, our review of the literature at the time suggested the high fertility phase of women's ovulatory cycles is accompanied by a moderate to possibly even large increase in their scent attractiveness as assessed by men, in their vocal pitch, which is a known correlate of vocal attractiveness, and also in certain aspects of their social behavior. So for example, their choice to wear more attractive or sexy or revealing clothing. In addition, our review suggested that there were at least some hints that men might respond differently to women at high fertility versus low fertility. For example, one very bold study tracked female lap dancers across their ovulatory cycles, and they found that the women in their study earned $100 more in tips per shift at high fertility versus low fertility. In addition, a couple of studies had shown that women reported that their male romantic partners were more possessive and jealous at high fertility versus low fertility. And so based on this evidence, we concluded that findings were at least consistent with the notion that men could detect ovulation, that they found high fertility cues as more attractive than low fertility cues, and that they might even behave differently towards women at high versus low fertility. So since our initial review, and perhaps in part because of the attention that it drew to this literature, a number of studies have been published examining various potentially detectable changes in women across the ovulatory cycle or fertility cues. And additional unpublished studies remain. Like the cycle shifts literature on mate preferences, these studies have varied widely in the methods that they've used to assess women's fertility across the cycle, and findings have been somewhat mixed. So again, this presents both the opportunity and the need for a careful and rigorous quantitative review. So as a second part of my dissertation, Marty Hazelton and I are currently in the process of conducting a second meta-analysis examining changes in women across the cycle that could serve as fertility cues, focusing mainly on the question of whether women are more attractive at high fertility versus low fertility. So I should mention that data collection is ongoing. Researchers are still sending us unpublished data, but I still wanted to share some preliminary findings with you all today. So many of the methods in this meta-analysis were the same as in the past meta-analysis, so I'm not going to go into as much detail this time. Importantly, in this analysis, the variables of interest were changes in women's attractiveness across the cycle, or in correlates of attractiveness. For example, as I mentioned before, vocal pitch is is a correlate of vocal attractiveness, or other widely studied possible cues of fertility. For example, changes in women's attractiveness enhancing behaviors, so the extent to which they worried about what to wear that day, how much makeup they wore, whether they chose to wear sexier clothes, etc., and also how flirtatious they were and how receptive they were to others' attempts to initiate romantic involvements with them. So far our sample includes 40 studies, and those studies contain 95 effects because many of these studies have looked at multiple possible fertility cues. So we're using the same methods in this analysis as the last analysis, But importantly, in this analysis, Hedges G refers to the extent to which women were rated as more attractive or some other variable at high fertility as compared to low fertility in standard deviation units. So we've run a number of different analyses, but I'll just present the key results for now. So first, we had 8 studies that were measuring other people's ratings of women's scent attractiveness or their physical attractiveness. And across those studies, we found an effect of about 0.18. So this suggests that women are being rated as about 0.2 of a standard deviation more attractive at high fertility versus low fertility. So I should note that some studies also looked at how attractive people rated women's walk and their dance, but we removed those studies from this particular analysis because we were interested in looking at aspects of women's attractiveness that aren't so much under their volitional control. When I added those other effects back in, this effect actually got larger, but I think that that could possibly reflect shifts in women's motivations across the cycle. So they might be trying to dance more attractively at high fertility than at low fertility. We also found that women rated themselves as significantly more attractive at high fertility than at low fertility, and this was a similar effect size. This could be veridical, so in other words, women's ratings of themselves could provide yet another window into actual changes in their attractiveness across the cycle, or it could reflect shifts in their motivation across the cycle. For example, women might just feel sexier at high fertility, and therefore they might rate themselves as more attractive at that time. We also found that women engaged in more attractiveness-enhancing behaviors at high fertility than at low fertility. Slightly larger effect, but still pretty small. So for example, women reported wearing more makeup, wearing sexier clothes, spending more time on their clothes and their makeup and their hair that day, etc. So it looks again like there's a shift in women's motivations here. And lastly, we found that women reported being more flirtatious at high fertility than at low fertility. They were also rated as being more flirtatious at high fertility than at low fertility, and they were more receptive to others' advances. So for example, a few of these studies looked at women's willingness to give out their phone number to an attractive male confederate who approached them on the street, their willingness to say yes to a dance with an attractive male confederate at a nightclub, or their flirtation with an attractive male confederate in an experiment. So you'll note that in all of those cases, the confederate was selected to be attractive. So it's unclear whether they would be flirtatious with just any guy at high fertility. It might be contingent on their having selected a very attractive confederate. So again, it's important to ask whether publication bias can account for the significant cycle shifts that we observed. Since data are still coming in and these results are so preliminary, I decided that for now I should probably just present the funnel plot for the focal analysis examining cycle shifts in women's attractiveness. And in this plot, there was just a hint of bias, and so we used the trim and fill procedure to impute one putative missing effect. Again, this had a negligible impact on the mean effect size, and so at least for now, it looks like publication bias cannot account for the significant cycle shift that we observed. So given this evidence for detectable cues of fertility across multiple channels, an interesting question is whether women's current romantic partners might be able to pick up on these cues at some level, and whether that could have measurable impact on their behavior. So a few years ago, Melissa Fallis got really interested in this question, and she wasn't just interested in whether men's behavior towards their partner would change, but also in whether men's behavior towards other men might change. So she had been reading about the challenge hypothesis, which has been supported by findings in a diverse range of different animal species, from birds to some of our closest non-human primate relatives. And the challenge hypothesis has found that in the presence of fertile females, males exhibit an accentuated increase in their testosterone when particularly threatening male rivals arrive on the scene. And so she wanted to see whether something similar would happen in men whose partners were currently in the high fertility phase of the ovulatory cycle. And so we designed a study, and we had 34 heterosexual couples come into the lab twice at high fertility and at low fertility. Again, the female partner completed luteinizing hormone tests to ensure that she ovulated near her high fertility session. And at both of these sessions, they completed a few tasks. So first, the couple completed a couple interaction task, which involved choosing a song and then slow dancing to it, taking a few cute coupley photos together, so that's one of our couples, and then finally having the male partner sniff his female partner's neck. And the point of all of these tasks was to ensure that all of the men in our study were exposed to a wide range of possible ovulation cues in their partner. So after that task, the couple split up and the male partner completed the rest of the study on his own.
They didn't actually split up.
Yeah. No, we broke them up. No. Right. So after that, the male partner immediately provided a sample of his saliva, which we later assayed for testosterone, and then they completed a competition manipulation. So for this task, they viewed 10 male profiles, and they were randomly assigned either to view 10 highly competitive male profiles or 10 more average male profiles. Each of these profiles consisted of a photograph and a self-description that had supposedly been written by the guy in the photograph, but actually were written by the researcher who designed the stimuli. So I'll give you an idea of what these looked like. So this guy goes on and on about how charismatic he is and how everybody's always trying to get him to try out for various leadership positions and blah, blah, blah. And he concludes by saying, we need some people who are good at making decisions. I guess I was made to be one of those people. So clearly he's the average guy. No, I'm just kidding. So he's the more competitive guy, and he was pre-rated as such. So we had 10 of those guys. And then here's another guy, and he comments instead on his modesty. And he says that some people are good at following. I guess I was made to be one of those people. So he's definitely less competitive, less threatening, more average seeming. So the guys looked at either 10 of the highly competitive guys or 10 of these more average guys. Then they completed one final saliva sample 15 minutes afterwards, so they had time for any impact of the manipulation to register in their saliva. And we also assayed that for testosterone. And then we looked at change in testosterone from before to after they had completed the manipulation. So although the fertility by condition interaction fell short of statistical significance, we did find a simple effect as predicted. So men who were exposed to the highly competitive male rivals did exhibit a more accentuated testosterone response to the manipulation in their partner's high fertility phase than in her low fertility phase, whereas men who were exposed to the more average rivals did not show any difference in their testosterone response to the manipulation across sessions. Okay, so I've presented a lot of different pieces of evidence today. Across all of these findings, I'd say evidence suggests that women's preferences when they're evaluating potential mates do vary systematically across the cycle and by relationship context. Their feelings about their partners also appear to change across the cycle dependent on their partner's characteristics. Men and women both seem to have the ability to detect cues of fertility in women, and they perceive cues of high fertility as more attractive than cues of low fertility. And the partners of women seem to actually respond to these changes in their partner by feeling differently towards other men at high fertility than at low fertility and experiencing a different hormone response to potential rivals. So in general, this work suggests that the ovulatory cycle might play an important but still often overlooked role in human social behavior, especially in the areas of attraction and romantic relationships. And this work has gotten a lot of attention from broad audiences, in part because it has potentially important implications for understanding attraction, dynamics of conflict and satisfaction and sexual desire in romantic relationships, male-male competitive behavior, or possibly even female-female competitive behavior, and also the role of scent communication in human attraction. So as this literature grows and matures, I've been noticing that it's testing increasingly nuanced hypotheses, which is good. It's also using increasingly sophisticated methods. So for example, the work that I presented just now suggests that meta-analysis may prove to be a very powerful and useful tool for synthesizing and evaluating sets of effects within this growing literature, especially given how methodologically heterogeneous it's been. But there are still many unanswered questions and issues to resolve in future research. So for one thing, our meta-analysis was not able to offer reliable estimates of cycle shifts in women's preferences for specific male characteristics, or to really provide an explanation of why the strength of women's cycle shifts in their preferences for these characteristics would vary by characteristic. So in the future, I hope that more high-quality research will be done to figure out this issue. Also, given that we found that women's satisfaction with their partners might shift across the ovulatory cycle, an interesting question is what happens then when women start using hormonal contraception, which wipes out these shifts in their hormones that probably drive these kinds of effects? Could this influence their relationship functioning? And do cycle shifts in women's feelings towards their partner usually have a cumulative effect on their relationship functioning? Another question that remains, so all of the findings that I presented today suggesting that women are more attractive at high fertility than at low fertility, or at least that others can detect changes, can't really differentiate between whether people possess adaptations for detecting fertility in women, so that they would actually detect changes across the cycle, or whether they have adaptations for detecting differences between women in overall condition. So if overall condition reflects underlying estrogen levels and estrogen levels differ across the cycle, then if people possess adaptations for detecting between women differences in estrogen levels, they might still rate women as more attractive at high fertility than at low fertility. But it wouldn't be accurate to say that they have adaptations for detecting fertility. So that's another issue to be resolved by future research. Another general question is how large and how meaningful are these effects in the real world? So both meta-analyses generally found smallish effects. They were robust, so I do think that they're real, but they were small. And that's based on a lot of studies that have been conducted under very, very tightly controlled lab conditions. So it's possible that once you take these things out into the real noisy world, these effects will be so small as to be meaningless. On the other hand, many of these studies, as I mentioned, use potentially problematic methods for categorizing women as high versus low fertility. So there could be a lot of error there in their categorizations of women as high versus low fertility, and that error could actually make the effect sizes that we've observed in lab studies be underestimates of the true effect size. So that's yet another question for future research. And finally, all of this research, as far as I'm aware, has been conducted on weirdos, so usually undergraduate women who experience many, many cycles in their adult lives. This is relatively unique in the world and is definitely different from our ancestors who spent most of their adult lives pregnant or lactating and actually probably experienced a few cycles while they were lactating. So how that would affect the size of these cycle shifts is a question that remains to be answered. It could make them more pronounced if a woman only experiences a few cycles in her adult life, or it could make them less pronounced. And I'm sure you can think of many, many more interesting questions, but I think I'm out of time for now. So thank you so much for your time and attention. I'd also like to thank my advisor and my collaborators, my fellow lab members, my totally awesome research assistants without whom I could not do this work. And all of you and Beck for having me today. Yeah.
So I was thinking, I like the P-curve analysis and all that, but it seems that if we one day soon have a world where p-values are no longer the gatekeepers for publication,
Right.
the criticism about shifting the window on the fertile phase in order to find the most sensitive window that you can find is
Right.
actually not really a valid criticism, because you probably would want to actually look at the posterior. You'd want to actually find out what is the best window to use, right? By looking at a bunch of data sets.
Right.
Rather than saying we're going to set it at 6, even though we have no idea if that's a good window.
It's probably not. Yeah.
Find ourselves to any other possibilities prior to looking at the data.
Right. Yeah.
And if
So
you
that's
had access to a lot of these, to the raw data, because your effect size is right. If you're using the researcher reported effect size using the window that they chose, you might also be misunderestimating the effect size. If you got access to all the raw data, put it in a giant regression, and didn't actually set a cost arbitrarily, you might be able to make a better estimate of these effect sizes.
Yeah, I totally agree, and I think that that's exactly what we should be doing. And we're actually working with Lisa Welling and David Putz and a few others right now. What we've done is we've combined data across many, many of these cycle studies, and we're using regressions to try to predict what are the best high and low fertility windows for categorizing women. Because I don't think it's realistic to expect researchers to start using the very rigorous lab-based methods that we tend to use in our lab. They're more expensive. You have to have women doing urine tests, and then you have issues with compliance. You can't possibly get a large sample of participants unless you have tons of research assistants, tons of lab space, and tons of time on your hands. And so I don't think it's realistic to expect that people are going to start using those methods in the future. So given that they're going to continue running these big online studies and then categorizing women or observations as high and low fertility, we need to find out which windows are best, and so we're working on doing that. We're also working on finding out rather counting forward versus back in the cycle is better. There's been a lot of disagreement about whether you should do one or the other, and so hopefully we'll have an empirically based guideline that researchers can use at some point in the future. And I think that would also help to really reduce concerns that critics of this literature have, that we're just trying out whatever and then, you know, reporting the ones that work. So yeah, it's a great point. Yeah.
So it seems like one assumption here about the high fertility versus low fertility changes is that it's based on this idea that I know that Marty and Elizabeth Helfer, the people have promoted about this dual mating strategy, right? That you've got this long-term partner who you're looking for to be the dad and your supportive, you know, provider, and then the short-term partner who is, you know, you're looking out for good genes, right? I don't disagree that that's a possibility. However, from my experience talking to women, it seems like another distinct possibility is that women are looking to trade up, right? And they may be trading up for a better genetic partner, or they may be trading up for a better provider, right? And so a lot of times what women will say is, yeah, I had an affair with this guy, and sometimes it's a long-term affair, and sometimes it's a short-term affair, but it may start out short-term because it's cheating, and then they wind up leaving the partner and moving on to this other person. And in that case, that seems to muddle this in that, so basically the idea that women have different reasons for committing infidelity, which often gets equated with being a short-term partnership. And that you wouldn't necessarily see the same effects if they were looking to trade up for another long-term partner.
Yeah, so in that case, are you saying that this could still account for the cycle shifts that we see in women's preferences? Because I wouldn't expect them to only be engaging in infidelity if it is for the purpose of switching relationship partners in the high fertility phase. I would think that it wouldn't matter where they were in their cycle.
Yeah, that's right. So I just think that the reasons for the switch that should be built in. I'm not disagreeing with the results.
I know. I'm just trying to work through
real, but maybe part of what's accounting for the small effect size is that there are these different motivations going into women's preferences, and so I know there's some work that looks at like their current relationship satisfaction and things like that, but it seems like more information about what's going on with the women and what exactly they would be seeking might account for why you're seeing bigger effects sometimes and smaller effects other times.
Yeah, one thing that I want to do in the future is just, so I can't get the kind of nuanced information out of the data that's currently out there that would allow me to test those kinds of ideas. But one thing I do want to do is compare the magnitude of these cycle shifts in women's mate preferences in single women versus women who are in a relationship and who are satisfied with their relationship. Because if they're satisfied with their relationship, they're going to stay in it. Then at least my guess would be that they're not looking to switch to a different partner. And so you might see then a different pattern of effects than single women who are engaging in short-term mating for another reason. But I don't know. I have to think about that.
One complexity in the possibility that Brooke raises is really hinges a lot on how much conscious access people have to their motivations. So it's possible that viewed at a distance, or even in the women's own minds in retrospect, they can see the trading up pattern, but at the time they may not experience it in those terms at all, right? So, here again, a lot hinges on when you define short-term versus long-term relationship partner. A lot hinges on people having some, conscious framing about what they're doing, and it's not clear that they necessarily should. So that again could account for small effect sizes.
Yeah, that's a great point. So I think one problem in this literature also is that a lot of researchers have asked women, like, how attractive is this guy for a relationship that won't last very long? And I think a lot of women are like, well, I don't want a relationship that doesn't last very long. I think that they should be asking, how sexually attractive is this guy to you right now? Because women might, choose to have sex with a guy because they think that he's going to be their partner. Not because they're thinking this is going to be a one-time thing. I think that'll make a huge difference, and so, yeah. So anyway, I'll definitely keep that in mind as I continue looking at these studies and see if anybody has data relevant to addressing those issues. Yeah.
So I'm glad you brought up the weird people point.
Yeah.
I think you can go a lot further with it than you did, and that it's not just the frequency of cycles. It's that we live in a zoo, right? That we don't get sick, we don't get injured, and we have enormous amounts of technology. I mean, the fact that you have to, you and your various colleagues in the field have to go to such great lengths to stop women from modifying their scent, for example. The reason that you need to do this is because we live in a zoo, right? That is, in the real world where people are, you know, exposed to injury and illness frequently, where they don't have the kind of technology to alter their appearance and their scent that we do, for example, you might expect these things to be much more dramatic. And that's, I mean, it's kind of a lesson from a lot of studies of animal behavior, is that what look like very small effects in the lab are much more dramatic effects when the animals are not buffered against actual injury and death and illness, and they're confronted with more real-world decisions.
Right.
Can I make a related one really quickly? So, I mean, I thought about when you were talking in the beginning about these measures of symmetry, right? So if this expressed symmetry is supposed to be a measure of how well these men or women or whoever it is have withstood insults to their immune system or something else, right? But if there are very few insults and very few stressors, which there are, related to what Dan is saying, right? Then you may just be measuring what their starting point is, as opposed to, or very close to what their starting point is, as opposed to it being an actual indicator of their ability to withstand stressors, right? So
So the idea is that the psychological mechanisms that women possess will still be sensitive to that variation, whether or not it currently reflects the kinds of insults that you've encountered during your development. But there'd probably be less variation in a population like us versus a population out in the Amazon or something like that. And in fact, some studies have found that preferences for more symmetrical partners are stronger the closer you get to the equator, and the greater disease prevalence there is. Yeah. Yeah.
This is kind of a basic foundational question. What is the accuracy of the LH population tests?
Oh, well, according to the companies that produce them, they say that they're 97% accurate in verifying, or in catching ovulation as verified by ultrasound. So that's the rate at which they correctly identify ovulation. I don't know what the rate is that they miss ovulation. Hopefully probably pretty low. I don't know if anyone else has thoughts on this. Some of
they're
mine.
very accurate.
I think they're quite accurate.
The gold standard method to use for this.
Yeah. Oh, sorry, go ahead.
Yeah, it just struck me that by focusing on basically slicing the cycle into the fertile days and the non-fertile days, and ignoring the fact that there's a very significant difference between the menstruating days and the non-menstruating days. In other words, you know, my personal experience is that the cycle shift is much bigger during menses than during the fertile period as far as behavior goes. And
You mean a shift in sexual desire, or?
well, okay, to just say this, sexual attractiveness, where you lump all the days other than the fertile days together, so you're lumping the infertile days in. You have a single woman who, during her the menstruating days, you know, you show them a picture of, you know, her ideal guy, and does she want an advance from him at that point in time where she would have to shut it down, or would it be a less attractive encounter for her?
Right.
You know, so it seems like that there's another cycle shift there that you I don't think that it's right to lump all of the non-fertile days together and just treat that as a single body of data, but you've got to see what the cycle shifts are during menses, because
Keeping it as a continuous variable would take care of that issue.
Yeah, well, not necessarily, because if there are other reasons why women's behavior is changing that are related to menstruation and not just related to the fact that their fertility is low, then that could actually still drive any correlation or differences between high and low fertility, even if you're using a continuous variable. But what I will say that I didn't mention, because I couldn't get into the details of all the different kinds of methods that have been used, is most studies either use a continuous fertility variable And so then there are only a few days when women are menstruating, and so hopefully that wouldn't drive any correlation that they see between fertility and whatever aspect of behavior they're looking at across the cycle. But another thing is, of those studies that have instead used the discrete high and low fertility windows, which is the vast majority of studies in this area, they usually eliminate premenstrual and menstrual days, so that they're comparing days in the late follicular phase of the cycle just before ovulation to days in the mid-luteal phase of the cycle. So I'm just sorry that I didn't say that starting out.
Yes, okay.
So they usually exclude those days for precisely the kinds of reasons that you were alluding to. Yeah, but great point, yeah.
So I was wondering, Kelly, if you had you said that you were looking into the idea about what's the best way to test ovulatory effects.
Mm-hmm.
I was wondering if you were willing to share that.
Oh, I don't know yet, actually, because I'm not the one running the analyses. Lisa Welling is, and so I'm not actually sure what the conclusions are at this point. One thing I would say, though, is if you're going to run a big online study, make sure that you limit the sample to women who report a very high level of confidence in how long their cycle is, how regular their cycle is, and when they last menstruated. Because a lot of the studies that made their way into my meta-analysis, for example, were studies in which they just asked women, when do you think you started your last menstrual period? People report it, and they're like, okay, then you're on day 5 of the cycle. But they didn't obtain any measure of how certain women were of that date. And past research has found, in studies where they ask women when they last started menstruating versus ask like a medical professional who had actually kept track of when they were last menstruating, that women are really off in their memory. They're not good at recalling. So I would say limit your study to women who are really sure of where they are in their cycle.
You can imagine that you could have a measure of conception risk, right?
Mm-hmm.
that borrows from sort of this well-studied and well-understood area of research in psychology, which is psychometrics, right? Where you ask the same question 10 different times to get at some, underlying construct, right?
Mm-hmm.
That there could be something like conception risk, where you can use multiple estimates too. Some people say 5 days, some people say 6 days. Well, use both of them, right? You can imagine there could be an argument for using both of them. Right?
Yeah.
And then something like some waiting for certainty about it. And then whether or not you have this lab data that could actually be added as a component, right? Something like that you can imagine could be, pretty.
Yeah. I can't imagine, and that's the goal, but unfortunately we're not quite there yet. But we should keep talking about this. Yeah, go ahead.
Yes, about the women were more attracted to, there was more of a cycle effect for the scent of men's symmetry than the other.
Yeah.
And I apologize if this sounds too vague, but it occurs to me that one of the things that's different about the scent of symmetry from all those other cues is that there's little or no socially transmitted ideas about what kind of scent is attractive. And I just, I think of this as maybe some women, so one of your other cues of behavioral dominance.
Yeah.
It could be that women are thinking, well, I shouldn't be attracted to a guy like that because maybe he's a jerk. Or it's, some sort of some sort of script.
But we don't have any
no
explicit ideas about what we're supposed to look for in somebody's body odor. At least I don't think so, not in this context. No, that's a great point. That's the first time anybody has proposed that as a possible explanation. I like it. So, yeah, it's great.
This is really interesting. I have a couple of questions, but I also, Joe, I wanted to just go off of what you just said and just raise the question of whether I would contest that there's no socially shareable cues about
Maybe intensity.
I think that's something people talk about. Certainly like within social circles that I have at one time in my life been in, is, if someone is smells like very young, that's something that is shared information. If someone smells really gross, that's something that's shared information. So I think there is some amount of social information that gets shared about attractiveness of male scent, at least within, probably certainly on university campuses where you have groups of people who all tend to know the same people.
Do you think it's specific,
campuses.
or do you think it's just he smelled good or he smelled gross?
Well, yeah, it's hard to describe the smell.
Because it's hard to that's not the same as he smelled like a combination of musk and the ocean or whatever. I don't know.
Hints of blackberry. Trouble. No, I don't know, but I mean, I think there's certainly some amount of information that would be I don't know, I just to put out the possibility that there is some amount of social discussion that
I
might go
wonder
on.
if in this kind of cultural context, like he smells bad just amounts to he smells a lot versus he smells good is just he doesn't smell like much of anything at all. But I don't know. I don't know if there are specific qualities aside from just intensity that are predicting whether people think that somebody smells bad. I don't know how high the consensus is either. But
The scent industry is a multi-billion-dollar industry. People are spending the money for some reason.
Yeah, so Brooke had there are a number of issues here. One point of Brooke's is, you presented scent of symmetry work in terms of cues of quality, which is different than compatibility,
Yeah.
right? So it could be the case that, as you already know from the work you've done with your collaborators, right, that these are idiosyncratic. Initially I was persuaded by Joe's objection, but listening to Erica, now that I think about it, and I think perhaps this is what you're saying just now, that is, even if we don't have the vocabulary for discussing scents, that doesn't mean there aren't cultural norms about it, and those norms are present in the form of the products that are being sold, right? So, there are, you can go to CVS and irritate the clerks by cracking open a whole bunch of bottles, and you'll find that there are a couple of sort of, themes, as it were, right? And those, even if we don't have a clear vocabulary for describing them in words, those provide a cultural norm about, what acceptable scents are.
Right.
So, the same is true, for, if you think about fashion or something like that, for someone who is not an avid consumer of the fashion industry, they might have a difficult time with vocabulary in terms
Right.
of describing
But they still know
different
like-
styles of, but they can, your participants can still look at those photographs and say, yeah, she's trying harder here than she is here.
But maybe less so with scent. It could be that they're not quite as explicit, or that we don't have quite as strong a sense of what's attractive in a scent. Then I don't know. It's an open question, but sorry, I don't know. Erica, do you want to go?
Sorry, can that was not actually
She
a
actually had a
question. I actually had two questions. One was about what the scale was on the sort of attractiveness ratings, because I felt like it didn't actually seem like people were rating them as more attractive.
Oh.
They was rating them as less unattractive.
That's-
You're correct.
Because I was really
Yeah.
surprised that they were all below 5 if these are very unattractive to very attractive.
Yeah, so I will say that, the scale went up to very pleasant and all the way down to very unpleasant, and most of the ratings were hovering around neutral to unpleasant, but they did use the entire range of the scale. So some people were circling 9, like that smells really great to me, and I have to say that a lot of the participants, especially the guys, while they were completing the task, would comment aloud on the scents and would occasionally be like, ooh, she smells healthy, yeah, and then circle them. So I tried to keep that to a minimum, but some people would still comment before I told them to please not talk out loud. So they were using the whole scale. Did you have a thought on that?
No, that just struck me as I was curious, all those seem to fall below what I assumed
Yeah.
was
I mean,
neutral.
Yeah, you're perfectly right. I mean, part of this might just be that there are these ideas that you're not supposed to like any body odor at all. So the best it can do is neutral, and then it can do a lot worse.
Yeah, run the study in Spain and you get the positive scale.
Yeah, I'd love to run these studies
So-
someplace else.
I have a really, really quick follow-up question. So,
Sure.
I was really interested in the fact that you found women's self-attractiveness ratings as varying as well, and I wanted to have anyone had done a mediation study to look at whether women, if you're getting the ratings from the same women, if women at points in their cycle where they find themselves less attractive or more attractive show correspondingly greater or lower shifts in their attractiveness ratings of other people. Because if you're in a period where you find yourself more attractive, then maybe you're going to rate other people as more attractive or less attractive. I mean, I'm just curious whether
I
you
see.
see a
So
relationship.
correspondence between your self-ratings of attractiveness and your mate preferences.
Right, because
that
I mean,
concept?
if you see yourself as a period where you're not very attractive, then you might not be competing for someone who is very different from you on the attractiveness scale.
Sure. Yeah. So I can't think of any studies that have been published looking at any such mediation. And I actually can't think of any data sets that have both measures of mate preferences and self-ratings of attractiveness, but I can think of a lot of studies that have ratings of self-ratings of attractiveness and then women's behavior on simulated shopping tasks where they could choose outfits that were more or less sexy. So I could actually get in contact with those researchers and say like, hey, do you find that women who are feeling sexier at high fertility also are the ones who are driving these effects where you see that they're choosing more attractive clothes at high fertility or something like that? But I couldn't probably quite test out the idea you're saying. Ideally, we would see correspondence between all of these different measures if they're being driven by shifts across the cycle. So, yeah, thank you.
I just want to come back to this issue of MHC compatibility because
Sure.
I just started digging into this literature, but it seems like there, at least from what I've been reading, that there's a lot more traction on the compatibility than there is on the general good genes. Now, that again may be cross-culturally variable in that in places where just general genetic quality is going to be really important,
Right.
then you may find more of a good genes kind of hypothesis working better. But I'm just wondering how the compatibility angle fits in with these shifts, and if people have done studies that are specific to where instead of just asking them to rate men in general, if you have measures of compatibility to see if they're shifting according to that.
So a few thoughts on that. One, I think in studies that have looked at who ends up with who in terms of like looking at married couples, they do tend to find that married couples are more MHC dissimilar than you'd expect by chance or adjusting for a number of other factors. As far as whether people tend to prefer the scent of more MHC dissimilar people, I think that evidence is very, very mixed. And then to the last part about cycle shifts and preferences for MHC compatibility, so one of the scent of symmetry studies also included a measure of preference for MHC dissimilar scents, and they didn't find it in that study. Also, there was one study looking at women's feelings about their partner as a function of their MHC compatibility with their partner across the cycle, and I think they found that women who are less MHC compatible with their partner showed, what was it? Do you know,
shifts in
Christina?
extra-pair attraction.
So they showed a larger shift in extra-pair attraction. So they're more attracted to other people if they and their partner are less similar. Is that correct?
More similar.
Yeah.
Sorry, more similar, less
Yeah.
compatible.
Yeah.
Yeah. Right, but I think that's the only other cycle shift study I'm aware of looking at MHC compatibility. I mean, I've always wondered how big we should expect those effects to be in a population like this, because again, like we're not really exposed to a lot of diseases or things growing up that would tell our body to prioritize MHC dissimilarity when we're choosing a mate. So
That should work equally well with the good genes
Yeah,
hypothesis.
I
agree.
I agree. Yeah. So I don't know. I don't know if that helps to answer your question. I guess I would just say there's kind of a dearth of evidence so far, and there's only one study that has kind of pitted the two together and didn't find it for compatibility, did find it for scent of symmetry.
To know if in general the MHC dissimilarity is if you pick two random UCLA students, for example, and look at their average MHC dissimilarity, if their dissimilarity is higher than it would be if you go to a small scale population, right, where there's
It has to be.
Oh,
exactly.
I
And
think-
so you may not be
think it
finding those
is.
effects because you're looking for them in this very varied kind of population.
Right, yeah, and I haven't done any research on MHC compatibility at all, so I don't know. Christina, do you have any thoughts or any? Sorry, it's just.
I think the studies that find, often the strongest effects of like MHC dissimilarity are like inbred.
Yeah, it's like the Hutterite study
Yeah.
is the biggest one. But there aren't very many the only natural fertility study I've ever seen is the Hutterite study.
Yeah.
So,
And really, it might just be that we don't need to have a preference for it just because by chance alone we're going to end up with a fairly dissimilar partner.
Right.
Because there are some theories that it's really about incest avoidance.
Well,
And it might just not be an indicator.
I mean, inbred is one way of describing it in terms of what the average degree of relatedness is. Another is that it's an endogamous community, and in small endogamous communities are our EEA. 20,000 people in an endless mating market who come from every population on the planet, which is basically your subjects. That's not our EEA. So this is what Brooke's point was, which
the effects will be washed out.
the effects will be washed out because if you just choose another individual at random, the likelihood that there will be substantial overlap is going to be very small, and the psychology shouldn't expect that to be the case. The psychology should expect there to be a quite reasonable chance of substantial overlap because populations were smaller and endogamous.
So what I'm saying is, so if they take a sample of university students and they look at the spectrum of that MHC dissimilarity, right, and they don't show an effect of dissimilarity, it may be that within the range of dissimilarity that is possible, they're looking at something, or sorry, similarity that's possible, they're looking at like this tiny window, and so maybe that's why they're not picking it up.
I think you're definitely right about that. And I know some of the data sets that Marty has where she's looking at MHC compatibility as a predictor of relationship satisfaction stuff, finds that most couples just have very, very low numbers of matches on MHC alleles, which is not, it's just not really necessarily the relevant population, yeah. And I know that at least one study on mice had manipulated whether the population of mice was exposed to hepatitis or not, and they found much stronger patterns of MHC dissociative mating amongst the mice that had been exposed to hepatitis. So it could be something that's like facultatively adjusted in response to cues from your environment, that you're in like a high pathogen load environment, something like that. But yeah, great question. Yeah, it's too bad I don't have a better answer for you. Okay, anything else? All right, thanks so